Okay, this lecture is about glycolysis, and this lecture is really focused towards like medical students. I know a lot of the audience that watches videos at this site is most interested in nutrition. I'm going to talk a little bit about nutrition, but this is really mostly from the biochemistry perspective of a medical student or anybody in a healthcare grad school taking biochemistry. I'm also going to talk a little bit about the philosophy and attitude towards school for academics. That's one thing I did. I used to do tutoring. I was really interested in academics in a major way. I didn't even plan my life this way. It just happened. I wish I was a wrestling coach. I would have had a lot more fun, but I just became like this monkish scholar, and it just happened. And at least I like being a scholar, but I'm going to talk pretty intense about what it takes to do really well in school, like to be the first in a medical school class. All right, so biochemistry is the most difficult subject in medical school. And the good news is if a person can learn biochemistry, just glycolysis is as difficult or more difficult than anything else in biochemistry. If you can learn glycolysis, this cycle we're about to talk about, you can do anything in medicine. Okay, intellectually, anything in medicine. And the first step to being good at school is to know what your goals are, why you're doing something, to have an optimal attitude. I've seen a tremendous number of kids, they pay these real expensive tuitions, and then they go to college, and they're not really sure what they want to study. They have really terrible study skills. They don't know what they're doing, and they're disappointed with their results. And so, you just have to accept the fact that regular schools do not care about you. They do not care about your education. If you find a teacher who cares about you as an individual, you are lucky. Okay, their job is to show up and babysit you for a couple hours. Okay, so your mom can go work and your dad. Okay, so what I'm talking about is something different, about real education, about real learning that's rather intense, and it comes from inside you. It's a personal thing. By the way, I'll just show you a couple books I wrote. I wrote Straight A's at Stanford and on to Harvard. I was student athlete of the year at Stanford, and it's like one of the best pre-med students in my group at there. I wrote another book on how to raise IQ and become a genius. I've read just about anything you could read on academics, memory skills, and study skills. I was really interested in this for a long time. And then I, raised my own kids. They're all grown up now, and, went through that with them. And also I did a lot of tutoring off and on through the years. Okay, so the first thing is you got to have your goals. If you just show up like the typical kid, I hope I do well, got to get a degree, get a job, make some money. That's an average way to think. You probably get average results. If you start thinking more intellectual, more open-minded, well, I want to try to learn what I need to know about biochemistry. That's much better. But I'm going to show you how to ramp it up to a higher level. In my mind, I was like, biochemistry is the language of God that he uses to write the book of life. How fortunate I am to have an opportunity to study this. That's a much better attitude because your brain is programmed to remember only stuff you really care about. If you say to yourself, well, I have to take this class and I want to get a good grade, you'll try to remember it on sort of like rote memorization for the test and you forget everything afterwards. But if you're like, this is the foundation of the knowledge that I want to have so that I can really understand medicine and really help people or really become good in my scientific field, that programs your brain, the importance of it to you to memorize it better, to understand it better. And at the college level and even the grad school level, most learning is largely memorization. It's only as you start getting more advanced, or if you're lucky enough to have a teacher who encourages it, that you start really doing thinking and innovating and stuff. But most college and grad school is primarily memorization. If you can go beyond that, that's fantastic. But memorization is what you absolutely have to be a great student at the college and grad school level. Okay, things like my father would say to me. He'd say, for all the recorded history, 95% or more people have been illiterate peasants and serfs, just trying to scratch a little food out of the ground so they don't starve to death. You're lucky to have the opportunity to study. One of the teachers I really like basically said something similar. You're so lucky to be able to study. When I was your age, I was drafted. My friends and me sent to the front line. Many did not come back. All you have to do is sit around and study. That's a good deal. And that was correct. I also had a conversation with my mother that I'll always remember when I was a freshman at Stanford. And I really didn't like it there because I was lonely from Chicago. I didn't know anybody. And I wanted to move back home to a college in Illinois. And my mother, I was closest to my mother than anybody else in the whole world, she said to me, Peter, And also I was frustrated because I got injured again wrestling, and I really wanted to be a wrestler. That's the only reason I went out there, to wrestle for the Schultz brothers. They're world and Olympic champion wrestlers. And so my mom said, Peter, I just want you to do your best for 1 year. If you don't want to stay after that, you can come home. The reason you got injured is because God doesn't want you to be a wrestler. He wants you to become a doctor or a scientist. You're going to be a great doctor or scientist. And I'm like, Mom, okay, I'll do it for you for 1 year, then I'm coming home. And I can remember, too, I looked out my window and, the kids had gone out to the parties, because most of the kids at Stanford were local from California or definitely West Coast, and they had friends, and they'd go home for the weekends. I'd be all alone in the dorms by myself. And so I see the vomit on the sidewalk. They're getting drunk and stuff. A lot of them hadn't drank that much in high school, and I'm kind of good. Glad to see you guys, screwing up and lazy. So even though you're a lot smarter than me, probably, you've had all these advanced classes. I never even took an AP class in high school. I didn't even know what they were. Both my parents were off the boat foreigners. I said, it doesn't matter, because it doesn't matter if you're smarter than me. When you're goofing off, I'm going to be studying, and I'm going to catch you. That was my determination when I was a freshman, and it was a good attitude to have. Freshman dorm at Stanford, there were 12 pre-meds, only 4 got into med school. Okay, a couple things about studying, and then we're going to get into the biochemistry cycle and the clinical applications of that. Get the old notes, because you want to go through the old notes. Pre-read as much as you can the night before, because that's what familiarizes you with the material, so you learn the vocabulary, you got the abbreviations so you can keep up in class. Your goal is to turn the lecture into a review. I usually would have 3 or 4 biochem books, and I would put them all out on a table. And what I was trying to do was, I would just stare at these biochemistry books. So here's a picture of the biochemistry cycle in one book, and I'd have other biochemistry books, and I'd open them all up, and I would just sit there and stare at it for a half hour, for an hour, whatever it took, and I would look at it and say, what is the irreducible minimum? How can I put this all together? What is the minimal amount of information I need to memorize from which I can derive everything else? And I would try to make a preliminary mnemonic at that point. I usually didn't know it well enough to make a good mnemonic. And the key was I was trying to turn the lecture, let's say it was the next day, I was trying to turn it into a review instead of being new material. Because I remember in the past, the biochemistry professors would go over too much material, and I would just be totally overwhelmed. So I wanted to get to the point where I could make it almost like a review. So I would study as much as possible in advance. And then I'd go to class, and it was almost like an emotional event for me. I was really psyched up for biochemistry because I knew it was the most difficult thing, and I was very interested in it. I had minored in chemistry initially, as an undergrad, and then I went to synthetic lab, and I hated the smell. It smelled like paint. So I'm like, I'm not doing this, even though I really thought chemistry was very interesting. So I was excited about biochemistry. And first of all, you want to write your notes in hand. You write much better. I know a lot of these kids type all this stuff now, but I think, you can draw structures and make arrows and all your abbreviations more freely. I prefer to write notes, and I think it connects to your brain better. I would try to study the notes as soon as I could after the class, because you could still hear the teacher's voice in your head, and you can fill in whatever gaps there are in your notes. Then after having this level of experience with the material, I really try to make a mnemonic that works. I've seen a lot of these lectures online, and most of these persons lecturing online are just sort of, they give you mnemonics that are too long. I want it to be concise, like an acronym. It should be as concise as possible. It's called chunking, to take a lot of material and compress it into something smaller from which you can derive the rest. Those are better mnemonics. I try to understand chemical structures to the extent that's relevant, because I know from experience that improves long-term memory. I like to be able to draw the whole cycle out on my own from memory. The mnemonic is the most important thing. I'll show you my mnemonic in a moment, and that'll really help you. I make illustrated condensed notes. I'll do practice questions in the question books once I've kind of got a better handle on the material. I was first in my class of 333 students in biochemistry and everything overall. And I had one of the highest biochem scores in the whole United States on my boards, maybe the highest. So I'm really used to pretty intense academic competition. Okay, now this slide is the most important slide in the whole talk from a medical student's point of view. So this is just the names of the substrates and products and some of the enzymes in glycolysis. There's about 10 steps overall. And the key point is, what's the unifying feature of glycolysis? And the unifying feature is the movement of the phosphate. If you can keep track of the phosphate, where there's phosphates on each of these different molecules, then it becomes easy to memorize the whole cycle. So what I did was I just made up a number to match the numbering of the phosphates, and then from that you can derive everything. Okay, trust me, if somebody who's taken this in med school, this is like a magic number for them. Once you've got this number, all the glycolysis will fall into place. And then it's good to have some sense of why the reactions are happening. So glucose in the extracellular space goes across the plasma membrane, enters the cell. Now it's in the cytoplasm. HK is the enzyme. Enzymes are in blue. So hexokinase will phosphorylate. By phosphorylating, you put a negative charge in there so it can't go back out through the membrane through the glucose transporter. And now it's also sort of energized. The next step is, phosphoglucose isomerase, where it's being converted, the glucose molecule. Glucose is an aldehyde. I remember it as glucose. That ends up being real useful. Glucose. Remember, it's an aldehyde. Gets converted into fructose, and fructose is a ketone. That's fructone. And the reason why it gets changed into fructose is because fructose is a more symmetric molecule, which is going to make it easier to split it in half into 3 carbon sugars. So glucose and fructose are 6 carbon sugars. And this number here, 666, is like the number of the devil. That's very easy to remember, okay? Then the next thing is, so here's the first 6, here's the next 6, fructose 6-phosphate, and then you're going to add another phosphate to the fructose, and that's going to be fructose 6-1-phosphate. Most people would write this out as fructose 1,6-diphosphate, but the reason why, or bisphosphate, the reason why I write it as 6-1 is so it goes with the mnemonic, 666. So there's 666, and then here's the 1. And then the asterisk is for dihydroxyacetone phosphate, which is given an asterisk because it doesn't have, its phosphate's not numbered. And that also is a helpful marker to show you that you're shifting into the 3-carbon phase of glycolysis. Now, the next thing to remember is the 3, and that's going to be for 3-phosphoglyceraldehyde. A key point here is I use lowercase a to remember aldehyde. That's going to be super useful. Trust me, you want to know that. That's going to make it so much easier to memorize the entire cycle. Lowercase a for aldehyde. All right, now we had a 1, 3, a 13, the bad luck number, which kind of goes after coming from the devil's number, if you will, to get to this point. And then there's another 13 here, and that's for 1,3-phosphoglycerate. ATE at the end of a molecule, its name, means it's a carboxylic acid that's deprotonated. So everything from here on out is going to be a deprotonated carboxylic acid. So capital A, 8, 8, 8, 8, 8. Okay, so you can remember that it's real easy. And now we knew that glucose was an aldehyde, so that's glucose. We know that 3-phosphoglyceraldehyde is also an aldehyde. We know that fructose is a ketone, so this other fructose is a ketone. DHAP is also a ketone. So these are the ketones here. And then it's all going to be carboxylic acids all the rest of the way through. So now you've got the basic idea of structure of these molecules. You don't need to know all the details, but you do benefit a lot from knowing that all these last molecules, 1, 2, 3, 4, 5 of them, are all carboxylic acids. That's going to help you a lot to remember the names of them and the reactions. Okay, what are some of the interesting things here? This 3-phosphoglyceraldehyde dehydrogenase is the enzyme. Super important. It's like, even though in school and for tests they're going to emphasize phosphofructokinase, that's fine. You got to learn that and how it's regulated based on the energy charge of the cell. If the cell needs energy, it goes. If the cell does not need energy, there's a lot of ATP available, then it's inhibited. But from a clinical point of view, 3-phosphoglyceraldehyde dehydrogenase, 3-PGA-DH, this is the enzyme that gets inhibited a lot, and it causes tons of clinical problems. This is the most important enzyme to be aware of. Some other things that are a little bit interesting. This one's called the mutase because the phosphate's moving around, but the structure of the molecule is not otherwise changed. This is enolase, this enzyme down here, and enolase is inhibited by F-. That's a little bit interesting. And then you end up with pyruvate. Pyruvate's the end product of glycolysis. But this mnemonic right here, that's the key. And pepper is just as PEP is for phospholinopyruvate. And PYR, pepper, PYR, that's for pyruvate. Knowing that mnemonic is the difference between crushing glycolysis and struggling with glycolysis. Just a little bit of stuff on terminology. When you have an asymmetric carbon, that's a chiral carbon, there have to be 4 different things attached to it. So for D-glyceraldehyde, this is the chiral carbon. Okay, this would not be because it's a double bond oxygen. It's a carbonyl group. When on your right-hand side, the hydroxyl group of this carbon, of the chiral carbon, is to the right, then it's a D. D for dexter. Dexter as in, dextrose, right hand. So that means it's a right-handed chirality. And again, it's an aldehyde, which means a carbonyl group with a hydrogen attached. That's actually really important and good to know. Here's DHAP, dihydroxyacetone. There's not a phosphate on it at this point. There's just a carbonyl group, meaning a double bond between carbon and oxygen. Okay. I remember that the sugars in the human body, almost all of them, are in the D form, meaning dextrose, the right-handed form. And I remember the word like a dog. Okay. D as in dexter, right-handed, because they've got the OH on this side for that chiral carbon, the last chiral carbon. O is the OH group, A for aldehyde here, and I'm just remembering D-glyceraldehyde. That's sort of the molecule I want to remember, because glycerol is like propane, triol. It's a real important molecule to remember, the 3-carbon molecule, because that's what you make fatty acids from, and that's what all the major pathologic reactions come out of this molecule, so it's worth knowing and remembering that they're D, and that's in contrast to amino acids. Amino acids are all L's, like the body, all these things have chirality, and the enzymes only recognize the proper chiral form in general. So that's worth knowing. Okay, this is just going through those same reactions again, getting to glyceraldehyde 3-phosphate, and then this is the key enzyme, glyceraldehyde 3-phosphate dehydrogenase. This is going to be inhibited by a whole bunch of different things, and once this is inhibited, you stop an enzyme, everything before it, you've blocked the stream, you've dammed the stream, and this is going to build up to very high amounts, and then it's going to start going through all these side reactions that are pathologic. Okay, so this is just showing some of the chemical structures written out, how glucose, the aldehyde right there, because there's just a hydrogen next to the carbonyl group. Ketone is buried inside of the molecule. There's a carbon on both sides of the carbonyl group, the double bond between carbon and oxygen. So I call it fructone. I call this glucose, and I call this fructone. Those little names will really help you to memorize what's happening. And like I said, it has to be made into fructose as its backbone structure, because that readily splits into 2 molecules of 3 carbon each. If you had stayed with glucose, it would have split into a 2-carbon and a 4-carbon molecule, and you wouldn't have been able to run this middle reaction here. And because normally, as these split into 3-carbon molecules, they're all going to eventually get made, all the DHAP's going to get made into glyceraldehyde 3-phosphate, and then that runs twice for every initial 6-carbon molecule here. You run these 3-carbon components twice. Okay, now we're going to show, after we get all the way, we go to Krebs cycle in the mitochondrial matrix, and then we're going to end up at electron transport in the mitochondria along the intermitochondrial membrane. So this is intermitochondrial membrane. Here's complex 1, complex 2, complex 3, complex 4. These are proteins working together to perform an enzyme function. And what they do is they hand off electrons sequentially from the weakest grabber of electrons, if you will, to the strongest grabber of electrons, which is oxygen. And as these electrons are passed down from complex to complex, a proton is pumped into the intramembranous space between the intermitochondrial membrane and the outer mitochondrial membrane. As pressure is built up in the intramembranous space, it creates an electrical gradient, which will then be harvested, electrochemical gradient, harvested by this transmembrane protein, which is an enzyme that makes ADP into ATP. So it'll add this phosphate to the ADP to make ATP, which is for the energy of the cell. It's like a $20 bill in a cell, that ATP energy. And this is also relevant because when this is overwhelmed, as it is in glycolysis what happens is too much fat enters the mitochondria, and it starts producing too many electron carriers like FADH2, and they overwhelm coenzyme Q and complex 3. And then everything stops and starts going backwards. And when it starts going backwards, coenzyme Q, instead of carrying electrons forward as it's supposed to, it'll start dropping electrons down to oxygen in the mitochondrial matrix. And that'll produce reactive oxygen species like superoxide. anion, which will have an unpaired electron in its outer orbital. And then that will be like a super ball bouncing around, breaking things. And it's going to set off a cascade of reactions that are going to go back to glycolysis and damage that one enzyme we just spoke about, glyceraldehyde 3-phosphoglyceraldehyde dehydrogenase. And that's sort of the money where most clinical disasters happen from diabetes. So here's that glyceraldehyde 3-phosphate. Here's glyceraldehyde 3-phosphate dehydrogenase, what we were talking about. And once this enzyme is blocked due to that cascade of reactions from the cause of insulin resistance, excessive fat entering the mitochondrial matrix, now this accumulates in great amounts. And when this accumulates in great amounts, it runs something called the PKC pathway, which is the protein kinase C pathway. And that's going to lead to what are called diacylglycerols, meaning, a triglyceride is like triacylglycerol, 3 fatty acids attached to that glycerol backbone. We talked about the 3-carbon glycerol backbone like a propane triol. And when that happens, that will activate protein kinase C, which is going to stop the glucose type 4 transporters from taking up insulin and taking up glucose into the cell. It causes insulin resistance, and that's going to lead to all kinds of problems. High blood glucose levels in the blood and everything that follows from that. In addition, some of this glyceraldehyde 3-phosphate is going to get made into what's called MGO, methylglyoxal. Methylglyoxal, it's like an alpha keto acid, and the point about it is it's going to go around glycating things and damaging things. So this, it's really important for a cell to be able to run these reactions without a problem. So a real high-fat diet messes up glycolysis and Krebs cycle and electron transport in the mitochondria along the inner mitochondrial membrane. It's a disaster. So that's why I'm not a big fan. You're going to hear all kinds of people telling you, oh, good fats. I'm not buying it, okay? All the fats will cause problems. Some people say it's especially saturated fat. I'm not going to get into all that stuff with fat. That's a topic for another day, but I'm just letting you know insulin resistance is a major problem for the human body. Okay, reversible inhibition of glycolysis by arsenate. So, the arsenic that has gotten into the food supply inhibits things. So, first of all, the insulin resistance leads to what's called a PARP enzyme that damages this enzyme in the setting of insulin resistance from a high-fat diet causing diabetes. Okay. Also, excessive drinking of alcohol will lead to accumulation of NADH, which is the product of this reaction, one of the products of this reaction, and that'll also cause a backup to this reaction. So excessive alcohol drinking will inhibit glycolysis. Okay, the next thing is arsenic. Arsenic will also inhibit this enzyme. The arsenic itself will mimic the shape of a phosphate, and it'll get attached to the 3-phosphoglycerate. So instead of forming the desired product, 1,3-bisphosphoglycerate, you're going to get this molecule with arsenic on there, and that won't be able to be used to make ATP. The word bis, when there's 2 phosphates, if they're attached to each other, like you remember ADP, that's 2 phosphates in a row, then it's diphosphate. But if the phosphates are on separate carbons, then they're called bis. So that means there's 2 of them, but they're on separate carbons. Now here's a bunch of other reactions that are activated once that 3-phosphoglyceraldehyde dehydrogenase is blocked and protein kinase C pathway is activated. You're going to inhibit nitric oxide, which is bad. That's what makes your vasodilator. That's the one Caldwell-Esselstyn talks about all the time. So you're going to narrow your arteries. That's vasoconstriction. You're going to have more platelet aggregation, more tendency to clot. So it's prothrombotic. And this is why diabetic patients are a vascular disaster. The most common reason people get their feet and their lower legs amputated. Every single hospital in the United States, every day, there's some patients getting their feet or their legs amputated because of diabetes. It's also so it's plugging up the arteries in their legs. It's plugging up the arteries in their eyes. It's plugging up the arteries in their brain and their heart and their kidneys. Diabetes is a disaster. The other thing, too, people talk about a lot of these rare diseases. Diabetes is so common in the United States that it's basically kind of thought of as normal aging. Same thing with hypertension. Okay, and they don't have to occur ever. All right, type 2 diabetes. All right, or hypertension. These are dietary diseases, and most people don't realize they're dietary diseases, which makes it harder for them to be able to help these patients. Okay, you'll increase endothelin, which is prothrombotic. Decrease the ability to clot for other reasons. There's problems with blood flow. We're not going to get into any more of this, but I just wanted to make the point. It starts with that 3-phosphoglyceraldehyde dehydrogenase, that glycolysis enzyme. Once that's inhibited, all kinds of things happen. This, by the way, is a Fisher projection, like a straight line, and for like a fishing pole. And this is glucose written out. It's got 6 carbons. So the first carbon is the carbonyl carbon. That's also going to be called the anomer carbon. It's D formed because 5th carbon, the farthest, most distal from the carbonyl group, is on the right side. So it's a D-glucose, as they always are. And the point is that most of the time in aqueous solution, which is in most locations in the body, the glucose is in a ring form. And this ring form can either have the anomer carbon OH going down. I remember that because alpha is shorter right here. So that's an alpha configuration. Or if it goes up, that's called beta configuration. I remember beta is taller, so the hydroxyl will go up. So the ring has to open, and it'll go into the Fisher projection, open chain, linear configuration, and then it'll reclose again in the other ring form. Normally, in the blood, for example, more than 99% of the glucose is in one of these ring forms. It just transiently opens up. And when it opens up, though, it can react with other molecules and form advanced glycation end products. So I did give a previous entire lecture about advanced glycation end products, but the point is this carbonyl group, of a glucoside and aldehyde is highly reactive, and it'll combine with an amino group on a side chain, like a lysine, for example, or an end amino group. And then it'll form advanced glycation end products like hemoglobin A1C, and that'll be an irreversible combining. That's why we call it end product. And that will damage not just hemoglobin, it can damage proteins all over the body. And we'll talk a little bit more about these advanced glycation end products, but not that much here. This was a topic of a recent lecture about a week ago or so. But the point I wanted to make was that was the idea of hyperglycemia forming advanced glycation end products in the blood. But inhibition of this enzyme, glyceraldehyde 3-phosphate dehydrogenase, is going to cause a backup of glyceraldehyde 3-phosphate, as we spoke about earlier. And that's going to then get made into MgO, methylglyoxal. Here's a methylglyoxal molecule, okay? And once that happens, it's going to react with things inside the cell to form AGEs, advanced glycation end products inside the cell. It's going to exit the cell and glycate things outside of the cell in the extracellular matrix, damage the collagen. It can bind to albumin in the blood, and that will then be taken up by cells by something called the RAGE receptor, which means the receptor for advanced glycation end products, and that will activate gene transcription to make a bunch of inflammatory molecules, inflammatory cytokines. Can lead to a cytokine storm-like effect, makes the platelets more prone to clotting, which can hide metastatic disease, increases cancer risk. It also causes a form of oxidative stress because it activates this enzyme system, NADPH oxidase, often called NOx. So I know I'm mentioning all this complex stuff. You don't need to know all this stuff, but what you need to know, though, is a whole cascade of harmful reactions is initiated by the initial high-fat diet causing insulin resistance within the mitochondria, initially of the skeletal muscle cells. So this is all the downstream effects. So what do? You just minimize your dietary fat and you never have to go through all this stuff. And like I said, this diabetes, insulin resistance is so common. People think of it as normal aging. It's not. I can tell you more often than not, when I look at a liver, it's a fatty liver. And that's kind of crazy. A lot of times, too, people are worried about rare diseases. Worry about diabetes and hypertension. If you can prevent those two or really minimize those two, you'll have prevented almost everything else. Diabetes is probably the most common cause of morbidity. Patients in the United States, and hypertension is not far behind. Okay, and you prevent both of those too, you'll probably prevent atherosclerosis, etc. You got a great chance to prevent dementia, etc. Okay, here's the second half of glycolysis. There's this sequence of reactions, and I mainly mentioned it on this slide for the purpose of showing you the enzyme enolase, and that's the one that's inhibited by F-, so that's why you want to have a reverse osmosis filter for your water, for example. Excuse me, or better yet, well water that don't have it. Okay, F- will also inhibit enzymes in the citric acid cycle. That's another reason why I don't want it in my toothpaste. It'll also inhibit complex 4, of the electron transport chain within the intermitochondrial membrane. So I just brush my teeth, every once in a while with a bare brush. I don't even use any toothpaste. I'm just trying to get biofilms off my teeth so bacteria can't live in them. But I don't sweat it too much. The main thing is I always floss at night because you got less saliva production at night. Okay, here's the last slide. And what it basically shows is the end product of glycolysis is pyruvate. When there's no oxygen available, it gets made into lactate. That's what's also happening in your cancer cells that are running anaerobically. It can go down the pathway of gluconeogenesis. It can go down the pathway of making alanine. It can go down the pathway of a step to become oxaloacetate in Krebs cycle. But most often it's going to go through this enzyme pyruvate dehydrogenase. Then it's going to get converted, decarboxylated, and it's going to be converted into acetyl-CoA, and it'll run into Krebs cycle in the mitochondria. But arsenic also blocks this right here. So arsenic can cause some pretty significant problems. So anyways, that's the overview of glycolysis. And if you remember that numerical mnemonic for where the phosphates are, everything becomes so much easier. And then I found it helpful also. Glucose is an aldehyde, glucose. Fructose is a ketone, fructone. 3-PGA with the little a is the aldehyde, and everything is an 8 after that, a carboxylic acid. Those little steps will enable you to memorize the whole thing in your head, to picture it in your head. And then once you memorize it in your head, just walk around and talk to yourself and say it all back to yourself. And then once you know it and you can reproduce it, you'll be so much more confident about it rather than, just worrying about your short-term memory to recognize it on a test. So anyways, hope that's helpful.